Hi, everybody. Sorry about class today and for the stuffy voice that you're going to get on this recording. Anyways, just like I said, wanted to stay on track, so this will provide us a way to do so. So covering chapter 18, looking now at controlling gene expression starting in prokaryotes, and the next time we'll look at eukaryotes. So assignment still chapter 18 due tomorrow by the end of the day. We'll start chapter 19 on Thursday, so those reading questions due before class. I did want to mention there's a schedule change. We're going to cover chapter 14 on genetics to go along with lab. So you'll see those extra assignments in so reading and also chapter assignments in eCampus, and also the new schedule, so not spending quite as much time covering some of the natural selection evolution chapters, so that it gives us some time to go through the genetics. Again, it'll just help us with our fly studies. And then also just a little bit more about the abstract. So the abstract is due this week in lab. It does have a separate rubric. It wasn't included in what was posted in lab 2, but it is posted and in lab 2. So whenever you go into lab and you click on lab 2, you'll see like the PDF, but you also see the abstract rubric. The abstract is really just kind of like a synopsis of the whole lab report. So it has a little bit of introductions, like the purpose, hypothesis, a little bit of the methods about how it was collected, a little bit of results, kind of like the main ideas, and then your conclusions that go in there. So again, little bits of each of those parts are what are provided in the abstract. So again, the rubric's posted to hopefully to help with what needs to go into that section. And then that is, I did have a question about whether, you had an abstract for like each experiment. Again, the lab report is as if you did everything for this. So it's not like divided by week 1 and week 2. It's just all one big lab report. So the abstract is for everything combined. But if you have questions about that, please reach out. All right, so learning objectives for chapter 18. So just looking at how gene expression can be regulated, talking about negative and positive controls, and some operon. So we're going to be looking at 2 examples, what's called a lac operon and a trip operon in bacteria. All right, so first of all, just gene expression, a little quick excuse me, review from transcription and translation. So cells selectively turn on or express genes, including when they're expressed and even if they're expressed. So again, all the cells in our body have the same DNA, but a cell in a muscle is going to turn on and make different proteins than a cell like a neuron that's in the brain. So the genes are there to make all of those, but again, we only want to turn on the genes that we need to make the proteins so that we have the correct phenotype for each cell type. Again, this is using a gene to make the RNA or protein. Excuse me. So controlling that expression is crucial. So we, our stomach cells have the ability to make hydrochloric acid, but we wouldn't want to make hydrochloric acid in our eyes. So again, controlling that, which proteins get made and when, is very important. And it's the same for bacteria as well. So we'll start with them if we understand kind of the general aspects of bacteria, then we'll move into eukaryotes next time. But we want to, effectively use our resources, again, making those so that if we make some that we don't need, then there are fewer resources to make the ones that we have to have. So again, we want to be able to control this. And then for bacteria, this is mainly going to be dependent on their environment, so what resources they have. It'll be a little bit different in eukaryotes when we get to them. All right, so how the bacteria control their gene expression. So again, information flow essentially in 3 steps, and this is the same for prokaryotes and eukaryotes. Of course, DNA goes to mRNA, this is transcription, then mRNA goes to protein as translation, and then even once the protein is made, it still has to be activated in some cases. So all 3 of these provide places for control. So we can control transcription, which is when the mRNA is made or if it is made. So the cell only makes mRNA that is needed to make the protein they need. Translational control, so not all mRNAs actually get translated, so we can control, even if the mRNA is there, it can be degraded more quickly, or it can be altered so that translation doesn't occur. Or there can even be post-translational control, so even once the protein is made, it may not be activated, so it can be chemically modified by adding like phosphate groups or some other groups to it to turn them on or turn them off. So if we look at transcriptional control first, so again, this would be blocking DNA going to mRNA. This is going to be kind of the slowest way of controlling the proteins in the cell, but it's definitely efficient. So again, this is going to occur before we expend any resources. So these regulatory proteins that are here would then affect RNA polymerase's ability to bind to the promoter, because that's what starts transcription. So if RNA polymerase can't attach to the promoter, then transcription can't occur. So that's how it would be controlled. For translation, a little bit more rapid changes. So this is going to have a little faster impact. But this is after mRNA has already been made. So we've already used some resources, but it can speed up mRNA degradation. So again, that poly A tail, again, chomping at the end, but we can increase how quickly the mRNA is broken down, and just the ability of the mRNA to be translated. So both of those could be affected, so that even if the mRNA is there, doesn't mean it has to be translated. And then the fastest response is post-translational, so kind of turning on or off the protein. So whether it's already been made, but is it actually functional or activated? And again, that's by those chemical modifications. So adding phosphates or acetyl groups or things like that, that could turn off the protein. So it's already there, but just because it's there doesn't mean it has to be functional. All right, and then just a quick little picture here to more easily visualize what's happening. So again, that transcriptional control is just controlling the ability of that RNA polymerase to bind to the promoter. Translational control, so again, here at the ribosome, being able to control whether the mRNA gets translated, and then again, affecting the activity of the protein by adding or removing phosphates or other chemical modifications. So now if we look at how we can control transcription, it can be controlled in a negative or positive manner. So if we look first at negative control, so some genes can undergo negative control, meaning that there's a regulatory protein called a repressor, and that's going to bind to the DNA, and as the name implies, it shuts down transcription. So here below, what we have is typically there would be transcription here, RNA polymerase attaches to the promoter, and we get transcription. However, if a repressor protein binds to the gene or to the promoter, that will prevent RNA polymerase. It can't actually transcribe it. So this would be an example of negative control. Then some genes also undergo what is referred to as positive control, to where instead of talking about a repressor, they actually need an activator in order to stimulate or trigger transcription. So under the idea of positive control, unless there is some type of activator protein present, there is no transcription. So RNA polymerase can't bind until this activator protein is present. If the activator protein attaches to RNA polymerase or to something, then and only then do we get transcription. So again, just kind of in general, some genes have this negative control where they are constantly being made unless a repressor is bound. Others, they only get expressed if they're activated to do so. So as we talk about some examples, we'll look at both of these types of control. All right, so the lac operon actually helps us to see an example of both because it uses positive and negative control. So this is a very commonly discussed gold standard model for looking at controlling gene expression in bacteria. So just a little bit about this lac operon and what are called operons. So in bacteria, these operons, which we do not have, but an operon is just a set of coordinately regulated genes that are transcribed into one long mRNA. So it's an mRNA molecule that is made up of multiple genes, and so it's called polycistronic. Again, this isn't something we see in eukaryotes, but prokaryotic cells do have them. So this example down here of a lac operon, there are actually 3 different genes, LAC-Z, LAC-Y, and LAC-A, and each one of those makes a different protein. However, instead of having a promoter at the beginning of each and every one of those genes, they have a promoter here at the beginning. So activation, so if RNA polymerase binds to this promoter, it is going to make an mRNA with all 3 of these genes. So it would make 3 different proteins at one time. And they're controlled or regulated by what's called an operator, which is shown here. So this would allow, this region of the DNA would allow control, so turning on or turning off transcription of all 3 of these genes. But again, they all get made at the same time. Now, on the lac operon, there's also a region here that allows for control. So in this, we have a LAC-I, and this makes a repressor that we'll talk more about. But here, there's a promoter and then a gene. So this is called monocistronic. It just, each gene is controlled by one promoter, and that's what we'll see in eukaryotes. But prokaryotes can have both. They can have some genes that have one promoter for every gene, or they can have this polycistronic example down here where multiple genes are controlled by one operator and they all get transcribed at the same time. And this lac operon is controlling the metabolism or the breakdown of lactose, hence the name lac operon. All right, so a little bit about the use of this. So in E. coli, for example, or in others, this lac operon, bacterial cells are going to use lactose only when glucose is depleted. So glucose is the preferred sugar of the cell. So if E. coli have glucose, they use it exclusively. But if they don't have glucose and they need to switch to lactose, they need a couple proteins to do that. So one of them for breaking down lactose is called galactoside permease. Excuse me, and that allows for transport of lactose. So down here at the bottom, so of course lactose is a hydrophilic molecule. It can't cross the membrane, so there needs to be a transporter to let lactose into the cell. Well, that transporter isn't always there. The only time that protein, that transporter, is present is if this protein, galactoside permease, is transcribed and translated, and then it's put into the membrane. So, of course, lactose has to be able to get into the cell, but then even if it gets in, another enzyme called beta-galactosidase, it is going to break down lactose into glucose and galactose. So this is called, or this beta-galactosidase enzyme down here, again, is a protein, so it has to be transcribed and translated to be present in the cell. And again, what it does is it brings in lactose, which is a disaccharide. So it's made up of 2 monosaccharides. So this enzyme, beta-galactosidase, breaks down lactose to give us glucose and galactose. And then those would go through glycolysis to get energy for the cell. Now, what happens, so as a way to control this, so beta-galactosidase is only going to be made if lactose is present. So again, it depends on the environment. So if lactose isn't present, there's no reason to make this enzyme to break it down. So lactose actually acts as an inducer molecule. And as we'll see here in a minute, inducers trigger transcription by removing a repressor. So again, a repressor would be bound, but if lactose is present, it binds to the repressor, gets it off of the operator, and allows transcription of the genes. And then again, because glucose is the preferred method, glucose, though, can negatively regulate the beta-galactosidase expression. So it's, as we'll see here in a minute, it has a way of inhibiting the transcription of this gene so that it doesn't make this enzyme to break lactose down. Because again, the cell wants to use glucose if it's available. All right, so if we look at this example here, kind of a negative control of the lac operon. So again, with negative control, it means that there is a repressor that is kind of constitutively or constantly made, and that repressor is always going to be attached to the operator to prevent transcription unless something removes it. So in this example with the lac operon, this is how it is. This is how it works. So the lac operon is under negative control. This repressor, this LAC-I gene, is constantly made, so this repressor is always present inside of the cell. So the repressor attaches to this lac operon and inhibits transcription unless there is an inducer molecule that is present. Okay, and the inducer, as we just mentioned, is lactose. So if lactose binds to the repressor, that removes or releases the repressor. So now we're going to get transcription of those remaining genes that are needed. So the beta-galactosidase to break down lactose, the permease that's going to let it into the cell. So we're going to get transcription of the lac operon when lactose is present. Again, the way the cell knows that is lactose binds to the repressor and it kicks it off. And so we call lactose an inducer. It induces the repressor to make it fall off to give us transcription. All right, now glucose, though, is also going to control. So it's not just the presence of lactose, it's also the absence of glucose that activates this lac operon. And it's an example of how it controls the operon and more in a positive manner. So what's going to happen is, again, glucose inhibits transcription of the operon because glucose is the preferred fuel source. If the cell has glucose, it doesn't care if lactose is there, it doesn't want to use lactose. So there are two ways that glucose actually prevents the lac operon from working or being transcribed. Here it's using an activator protein. So again, this is positive regulation, where the only time you get transcription is if an activator is there. So the activator here is called a CAP, so catabolite activator protein. So this provides positive control. So again, it causes transcription to occur of this operon. Now the only way, though, that this activator works is if cyclic AMP is attached to it. So cyclic AMP binds to this activator protein, and that allows the activator to attach and transcription to occur. Well, if glucose is high, if the cell has a lot of glucose, the way it knows that is it causes cyclic AMP levels to be low. And if cyclic AMP levels are low, then this activator protein won't attach because there's nothing there to activate it. Now, glucose also excludes the inducer. So remember, the inducer is what attaches to the repressor to get it to come off. So the way it does that is glucose inhibits the transport of other sugars. So if glucose is present, we're not going to be bringing other sugars into the cell. So what it does is it inhibits lactose transport. So if lactose can't come into the cell, then the cell doesn't have that inducer and the repressor remains attached. All right, so a nice little picture here to look at this. So again, with the lac operon, there's both an example of positive and negative control of this operon. So the cap protein is that positive regulation. So again, only whenever the activator is present do we get transcription. So if glucose is high outside of the cell, then that cyclic AMP is going to be low inside. And that's going to prevent this cap protein from attaching and activating the operon. So transcription is going to be infrequent because there's not that positive activation of the operon. If glucose levels though drop, now the cyclic AMP increases inside of the cell. Cyclic AMP and cap, that activator protein attached, that allows the cap to bind to the RNA polymerase, and that increases transcription. So that's positive control. Only transcription when the activator is present. And again, that is controlled by the levels of glucose. All right, now, again, with a negative control, this means that the repressor is, that means that the repressor is going to be attached unless the inducer is present to remove it. So this is negative control. Now, how glucose works with this is if glucose levels are high, that's going to prevent lactose from coming into the cell. So it inhibits transport. So if lactose, even if lactose is outside of the cell, it doesn't come in. So that repressor is going to remain bound and there won't be transcription. Now, if glucose levels drop, now we do get transport of lactose into the cell. Lactose is that inducer that binds to the repressor, and now it comes off and we get transcription. So transcription of the lac operon is going to be highest when glucose is absent and lactose is present. And that's going to allow the activator protein to attach and the repressor to be removed. Again, there are quite a few videos. I'll post a couple of links just to kind of visualize this if it helps. If again, you're kind of more visual, I'll post a couple of videos that go through this in addition to what we just talked about. All right, then another example here, kind of a twist on that negative feedback, is the TRP operon. In this operon, again, multiple genes are made, or multiple genes are controlled by one promoter, and these are all involved in making the amino acid tryptophan. Okay, and this, in order for the cell to make tryptophan, there are actually 5 enzymes that are needed. So all 5 of those genes are controlled by one operon. So it's called the TRP operon. They're controlled by one promoter. So it'll make all 5 of these genes at the same time. And again, that's called a polycistronic mRNA. Now, how this works is that the operator, again, that controls or regulates transcription, it overlaps the promoter. So we use a repressor protein. So this is that negative feedback, or sorry, this negative control. So the only time we want those genes to be expressed is if we need tryptophan. Okay, if there's already plenty of tryptophan in the cell, we don't need to or the cell doesn't need to activate this TRP operon. So the cell needs a way to know when tryptophan is present and when it's absent, so it knows whether to make it or not. All right, so this is an example of using that negative feedback. And we've talked about negative feedback before when we were looking at enzymes. And with negative feedback, that final product is going to inhibit further production of the product. So we talked about how, again, the final product would be an allosteric regulator of the first enzyme involved. Right, so this TRP repressor, it binds to the operator only when it's bound to its regulator. which in this case is tryptophan. So this is different than what we just looked at with the lac operon. So with the lac operon, the repressor is always present unless the inducer attaches and removes it. The trip operon is an example of when the repressor only binds when a regulatory molecule is attached. And in this case, that regulatory molecule is tryptophan. So when tryptophan levels are low, the repressor doesn't bind to the operator and we get transcription. Because again, the purpose of this is to make tryptophan. So if we don't have a lot of tryptophan, it needs to activate transcription. So then those genes are made and the mRNA is translated and tryptophan is now made. So as tryptophan increases, then it binds to the repressor. It's called a co-repressor. It binds and now the repressor changes shape and it is able now to bind and prevent transcription. And so back to kind of our enzymes, this is how allosteric enzymes work. So remember the allosteric, a regulatory molecule, in this case tryptophan, attaches to a protein and changes its shape. Okay, we just have a few different words here. So again, co-repressor, in this case, is a molecule that binds to the repressor and it is going to allow it to attach. The inducer that we mentioned with the lac operon, it binds and removes it from the operator. So again, here with tryptophan, we're still using negative feedback, or we're still using negative control because we're talking about the repressor. It's just this is a different example. Here, the repressor is only going to attach if that co-repressor is there. If tryptophan isn't present, if that co-repressor isn't there, then the repressor doesn't bind and transcription is going to occur. Again, with the lac operon, the repressor is always attached unless something binds to it to remove it. And so that's called an inducer. Right? So again, just a couple different examples because all genes can be regulated in different ways. This is just showing a variety of ways to control gene expression by co-repressors, inducers, or activator proteins. And then understanding these will help us whenever we move into the eukaryotic cells.